It's a pleasure to introduce Professor Kurt Callan. Professor Callan is a physicist by training with a bachelor degree from Haverford College in the US and a PhD in physics from Princeton University, also in the US. Professor Callan is currently the James McDonnell Distinguished University Professor in Physics at Princeton University. After a longer career working on quantum field theory, particle physics, and string theory, and also after many important prizes you can check online, Professor Callan shifted his research to cellular biology using modeling and statistical inference approach to deal with the extensive generated data in modern biology field. He works in problems ranging from the gene regulation in bacteria to the functioning of immune system in humans. Thank you very much, Professor Kurtz, for accepting our invitation, and so help me to welcome him.
So, can everybody hear me more or less? Okay, so I don't know you at the moment, so I'm not sure exactly whether I've pitched things in the right fashion for you. I'm going to treat this like I would a kind of upper undergraduate or first year or so graduate course in biology for physicists. And I'm going to try to pick out a few highlights of what physical thinking has to offer in thinking about biological entities. All right, so, and I'm going to, as I say, I'm going to treat it like I would a class in the sense that I will lift up the hood instead of showing lots of PowerPoint slides with nice pictures and an occasional equation that you don't have time enough to actually absorb. I'm going to actually show you how you derive things. All right, so it'll go slowly, but I think that has advantages. I think it's a good thing. You have to go through these things once in great detail. All right, so I've prepared a sort of modular approach. I'm going to talk about, once I find a piece of chalk, I'm going to talk about noise and diffusion in the life of bacteria, well, in the life of cells. And I've never given this precise kind of class before, so it'll be a little bit, how should I put it, scratchy. I will have to hop back and forth from one thing to the next. I'm assuming that everybody has a basic undergraduate level knowledge of physics. You know about diffusion, you know about the Boltzmann equation, et cetera, et cetera. Do I find, okay, well, all right. So if that's, how shall I put it? If I say something that doesn't correspond to something you feel comfortable with, speak up and I'll try to expand as far as I can. All right, so that's number one. Whoa. The second module is going to be on how do I deal with that? Is it the microphone? Is it this? Should I turn down the volume? Can I turn down the volume? Yeah, okay. That's one of those things. Okay, so then the next thing is going to be accuracy. You know that you reproduce, how should I put it, cells have a genome in the DNA molecules that record all of the information they need to live, and cells divide and multiply, and when they divide, they have to copy their genome. And there is, in fact, from the point of view of physics, a very profound problem. It's very important that the copying be exact. And we will see that there's a deep problem with that. And so how do you make things copy themselves with great fidelity? And the third topic is going to be, if you will, something about evolution. Thank you. And maybe, the thing that I'm really most interested in these days is how the immune system works. And the running, you want to change?
Can I just adjust
Sure.
the microphone?
Yeah, don't know. Where's best to put it?
I guess it's over here.
Okay, all right. And there's a running thread through all of these, namely that there's a problem that can be stated in mathematical language, and you can address this mathematical problem in ways that tell you something about how the biological systems actually work. Okay, now, there's a lot to say, and I probably won't get through all of this, but maybe that's not a bad idea. Okay, I'll go to in as much detail as we need. All right, so let's talk about number one. So to give you some context, so we're going to be talking about a famous paper. And once I get my bearings, I'll put these resources that are important for understanding what I'm going to talk about up on Dropbox, and in some fashion it'll be possible for you to download it, okay? All right, so famous paper number one is Berg and Purcell, and the year is something like 1974. In fact, maybe I can bring it up. Let's see if I bring it up. Oh, I'm in Zoom, right? Okay. Well, never mind. All right, so okay. All right. Okay, so we're going to go to documents. We're going to go to books. We're going to go to SAIFR. We're going to go to papers. We're going to go to Purcell. So this is the one. So this is called The Physics of Chemoreception. And the question that they posed was, how do bacteria find their food? Or how do they stay away from things that would be bad for them? And so the idea is that because bacteria are so small, the phenomenon of diffusion plays a big role in the ability of bacteria to find sources of food or to avoid sources of trouble for them. Now, so the phenomenon, therefore, in the end, is going to be diffusion and the physics content is going to be about diffusion and random walks in the particular context of cells trying to go about their business. And so there's actually several contexts, several versions of this problem. So at all levels, cells need either to sense or to capture from the environment individual molecules, all right? So if you're an E. coli cell and there's a source of glucose around, you will want to capture those glucose molecules and take them inside the cell. So the cell has, again, here we go. So there's a cartoon that I'm going to be using frequently, and the cartoon is this. So here's a cell. Suppose it's an E. coli cell, and it will have on its surface proteins whose structure is such that they are able to bind certain classes of molecules. So these are, these are sugars, things like that. So they're not very big, right? They're things that are made out of a dozen atoms or so, and they've got, their size is tens of nanometers. And these things are these receptor proteins. They're much bigger, but they will have receptor pockets on them which serve to bind specific molecules of this type. So they're designed to capture this kind but not that kind, and that has to do with a binding energy. It's strong for one of them and it's not so strong or non-existent for the other. So this phenomenon of proteins whose job in life it is to recognize molecules and either simply say it's there or actually to take it and ship it inside the cell so that it can be used, that's a rather common theme. And another version of it that we'll come back to from time to time is inside the cell. If we blow it up, we have the DNA. The DNA has code for some number of proteins, and that's of course how the cell does its business. So here is the DNA stretch that codes for a particular protein, like one of these proteins that winds up being expressed on the surface of the cell and serving to tell the cell what's going on in the world around it, okay? And in order to function, the DNA code for that protein has got to be expressed, okay? So there's a whole mechanism that reads the DNA sequence of a protein, turns it into RNA. Everybody knows about DNA, RNA, protein these days with everything we've learned about vaccination against COVID and other such nastinesses. And in the chromosome there are, let's see, for us there are about 20,000 different proteins that can be read out in this way. And you don't want to be reading out all of them all the time, right? The difference between a bone cell and a blood cell and a nerve cell is in which subset of all of the proteins that could be made are actually being made. So there's a short region called the promoter, sort of upstream of the DNA sequence for the protein. And there are other proteins, and so this whole dance is proteins talking to DNA, talking to RNA. It can be very complicated, but with equations we can sort out what's possible, all right? So there are things called transcription factors. And they're little proteins of their own, and they are made reading out genetic information on the DNA, to be sure, but let's imagine that they're there. And what do? Well, they are designed so as to bind to this so-called promoter region, and when they do so, they authorize the reading out of the information for the protein. So this is, again, protein binding to DNA. That turns on the reading out of particular genes, and so, how shall I put it? Systems biology, if you like, is all about how it is this dance of transcription factors being authorized to bind to their target sequences on the DNA, binding to it, and that's a question of binding energy, that is, or a cell sensing what's going on in the environment around it by molecules binding to these surface receptors. Okay, so it's a common theme. Okay, so let's suppose that we're an E. coli cell, and our goal is to eat some nourishment, some kind of sugar. Okay, so what is the picture that we have in mind? So once again, here's our little E. coli, and it's got its complement of cells, of surface receptors, and it is surrounded by some concentration of, there's some number of molecules of interest per cubic whatever, cubic centimeter per liter, whatever. So and this is usually expressed in molar, right? So this might be 1 micromolar, 1 nanomolar, 1 millimolar, who knows, of something of interest. Let's suppose it's glucose. Now, these guys will, as these guys wander around, so they undergo a random walk, and eventually any one of them has some chance of winding up, so to say, in the immediate vicinity of one of these receptors and being bound. And then if we're talking about eating, the molecules that are captured by these receptors are immediately internalized for the use of the cell. Now, the problem is that as they internalize the molecules that are of interest to them, they're essentially cleaning out the neighborhood, all right? And there's nothing more to eat. But these molecules are dancing around, and they will eventually diffuse back into the neighborhood. So there's some time scale on which this all happens. If they're very good at eating, they will clean out the neighborhood of useful molecules, but diffusion will bring them back in, all right? And so there's a time scale involved in this process, which has to do with the physics of diffusion. All right, so now we're going to talk about how fast the cell can eat the concentration of interesting molecules that are around it. Well, that's a problem in diffusion. So we're going to do the typical physics thing. We're going to say, let's make our cell particularly simple. We'll assume that it's a sphere of some radius. All right? This is the physicist version of a cell. And then there is some concentration of the things of interest. And so it has some dependence on where you are. And we'll find out more about that in detail. But when you get very far away, the world has supplied some concentration of these things of interest far away from the cell. And it's busily eating away at the ones that are near it. And we want to find out what is the rate at which molecules of interest are essentially falling on this model of the cell and being eaten. Okay? That's the problem. How fast can this guy eat? What is the rate of gobbling up? Well, that's governed by diffusion. So now we have to write down our first equation. All right? Actually, I want to make this radius A. Okay, so there is something called the diffusion equation, which writes as follows. Which says that the D'Alembert, a certain natural constant called the diffusion constant times the D'Alembertian of C is equal to the time rate of change. So the idea is that as this guy eats molecules that are close to it, it's causing a depletion of the molecules near it, and it's making a gradient. And when there's a gradient, there's a flow. And basically, this is saying that the flow is the diffusion constant times the gradient of C. It's the idea. We want to consider a situation in which the concentration of the thing of interest varies from point to point. That's the whole idea. We want this guy to be eating stuff that falls on it. It's going to essentially chew out a hole in the density distribution around it. And as a result of that hole, there's a variation from point to point. And when there's a variation from point to point of the concentration, this the random walking of the individual molecules macroscopically becomes simply a flow. There's a flux. Yeah? I can hear you. Oh, okay. Very good.
Hi. Oh my God. Okay. Hey, I'm not a physicist, and I don't know what is this triangle upside down.
All right. Excellent. Okay. So, but derivatives don't scare you, right? Derivatives are fine. Okay. All right, so what do I mean by del is just ddx, ddy, ddz. In other words, C is a function of where you are. Concentration varies from point to point, and you can differentiate the x coordinate, the y coordinate, or the z coordinate. Okay, and this is just the way you calculate the gradient. So the gradient of C is, it's a vector. You're happy with vectors. Good. All right, so the x component is the derivative of C with respect to x. The y component is the derivative with respect to y, and the z component is the derivative with respect to z. That's all. Now, what's del squared? Okay, so del squared is of something is just okay? It's the sum of the second derivatives in the three different Cartesian directions. Okay? Why? Okay, so how does this arise? Okay, so you're happy with this much, let's say. Let's see. I'm trying to think where to go next with this. Okay, so this is the basic thing. It says there's some empirical constant called the diffusion constant, and it has the dimensions of length squared divided by time. So the dimensions of d are divided by time, okay? And, one of the things I should tell you, what is a typical value of d? Microns squared per second or something like that. So the numerical value will be important, but it's just telling you about the mean square displacement in some period of time, and it has these dimensions. So what's happening here is that we're making use, to get from here, the flux, the rate in which molecules are flowing like water flowing past some gauging station in a river. There's a certain number of molecules per square meter per second. That's flux flow, okay? That's what this is calculating for me. And so J, X, Jy, and Jz, according to this, are d times dc dx, d times dc, ooh, let's continue on the next line, dc dy, d times dc dz. So it would be more convenient if we lived in one dimension. Then I wouldn't have to write all these vector components, okay? But we live in three, and we have to somehow live with that, okay? So this is telling me if there's a spatial variation in the concentration, then because of the random motion of the random walking of the molecules, there must be a flux, there must be a flow. And the idea is that if I have a high concentration here and a low concentration there, then the net effect of diffusion is going to be even it out. In order for it to even out, somebody has to come from here and go there, all right? And because the concentration is higher here, the number of guys going that way will be bigger than the number of guys going from the low concentration region that way, and we're going to find ourselves evening out, all right? And what this equation says is that the molecules never disappear, they're conserved. So there's a conservation equation, and the conservation equation is that del dot J is equal to dC dt. That's just saying the rate at which the concentration is going up or down at your place is the sum of all of the flows into that place from the different directions, the x direction, the y direction, and the z direction. And this dot notation, okay, those of you who took undergraduate physics, you know what it means, but it's simply saying it's just taking its ddx of jx plus ddy of jy plus ddz of jz. All right, it's taking this vector quantity and turning it into a number, a scalar. All right, and this scalar is just telling you how rapidly stuff is accumulating at the point that you're looking at. Stuff is coming in from the left, the right, the top, the bottom, and that these derivatives of the fluxes are telling you what's the difference between the flux to the left and the flux to the right. That's what the derivative x is saying. From top to bottom, that's this guy. From back to front, that's this guy. You add them all together and you get the rate at which the concentration is increasing. Okay, so this is a very quick tour of diffusion and the diffusion equation. But you have to have some feeling for the diffusion equation in order to understand how things live in the microscopic world. Okay, this is something that diffusion plays a big role in phenomena that occur in our world at the scale of microns, the actual size of cells. Okay, so what is this doing for us? All right, so we have this famous diffusion equation. And so basically all I need is this one. And maybe we just have to remember that this guy has the dimensions of length squared divided by time, and a typical number is going to be 10 to the minus, is going to be twiddles 1-ish microns squared per second, all right? You know what a micron is? The micrometer, 10 to the minus 4 centimeters, like 10 to the 4 angstrom, so it's like 10,000 hydrogen atoms stuck one next to the other. This is the scale on which things have to happen for cells. Okay, so I want to get back to the question of how fast an E. coli can eat. All right, so in the case at hand, so for the spherical cell, and in the end, it's not going to matter what its shape is. What's going on? Well, in fact, the concentration is going to depend on where you are, but the only thing that matters if we're talking about something that has spherical symmetry is that this guy depends on how far you are. So here's R. Here's my spherical cell. Here's the center. Here's how far I am from the center. And I'm assuming that when all the dust settles, the concentration is really only going to depend on how far I am from the center. Okay, so if you've done an undergraduate physics course, you're given some general law, and then you're said, but let's look at a really simple situation. Let's suppose we have perfect spherical symmetry, right? Suppose that the Earth is a perfect sphere, and then the gravitational force at some distance, at some point outside the Earth, It doesn't depend on latitude and longitude. It only depends on how far you are from the center of the Earth, that sort of thing. So we're making that nice approximation here. So this is the other trick you learn in undergraduate physics is changing coordinate systems. Cartesian coordinates, x, y, and z, for getting around in 3 dimensions are the easiest and most, well, they're not the most convenient necessarily, but they're the easiest to grasp. But in certain circumstances, it makes more sense to use a different set of coordinates. You know, when you're on the surface of the Earth, you don't use x, y, and z. You use latitude and longitude, and I'm at sea level, right? So I get around using angles rather than distances. So you can go from x, y, z as markers for where you are to a different coordinate system, so-called spherical coordinates. All right, so that's what I'm going to do because in my physical setup here, this thing which depends upon position only depends on how far I am from the center. It doesn't depend on angles. All right, so I would have to tell you how to convert this equation into this new coordinate system. All right? I would have to do that in order to, so what I want to do is to solve the equation. Okay, so what do I want to solve, actually? Excuse me, I should say that. So here I'm saying things can depend on time, and if they do depend on time, that has an impact on what equation I'm trying to solve. Okay, if I put the cell in its sugar solution and it's eating away, eventually things will settle down to, at any given point, nothing is changing. Things will settle down into what's called steady state, okay? So eventually we will get to steady state. Stuff just flows in at whatever rate it has to from far away, and it flows towards the cell, and the cell eats it as fast as it can. And my concentration, which initially I said could depend upon how far away from the center I am and the time, eventually it'll settle down to it depends only on R. It's in steady state, okay? And therefore the equation that I have to solve is that this d squared dx squared plus d squared dy squared plus d squared dz squared acting on, so that's the diffusion equation to solve, because d dt is nothing because we're in steady state. Nobody's changing with time. So the right-hand side of this equation is 0. And then, well, I'm multiplying by d, okay, but I'm just, the real condition is that this funny thing called the Laplacian acting on c is 0. That's my equation. And I said this del squared, this Laplacian, is d squared dx squared plus d squared dy squared plus d squared dz. Okay, and r, okay, well, r is actually just if x, y, and z are my coordinates. So I just have to solve that equation using the fact that r is this construct out of the x, y, and z coordinates. Now, it's much easier, much simpler, much more efficient to actually convert this Laplacian from its expression in terms of the Cartesian coordinates to something that depends, that explicitly uses only the spherical coordinates, all right? You can do this. I do not have the time to do this now, but I think you might want to try it. So in spherical coordinates, if I'm acting on something that depends only on the distance, only on r, then the equation actually reads, or something like that. Nathan, do I have it upside down? Nathan will not confirm or deny, but you can actually, oh, I'm sorry. Now I have it right. Okay, now proving that this thing is the spherical coordinate translation of this Cartesian thing, d squared dx squared plus d squared dy squared plus d squared dz squared, you know, this is something that you'll spend a whole day on in an actual undergraduate course, okay? And you'll go home and you do homework assignments and blah, and blah, and blah. So I don't have time to do that, but I ask you to trust me, but you should verify, okay? Trust but verify. So it's pretty clear what the solution of this equation is going to be. A constant plus something divided by r. So we're actually solving the diffusion equation in an interesting condition. So why is that? Let's see. So I should be able to add a constant to this thing. There's something wrong about what I'm writing, okay? Sorry? 1 over r. Yes. Thank you. Yes, okay. You can never work alone, right? Because when you get hung up, what?
I think you're in cylindrical or spherical. You're in spherical coordinates.
I'm in spherical coordinates.
So I think it's squared.
R squared was right.
Yes.
Okay, so all of a sudden, okay, so what you see right away is that that's a solution, right? Because if I multiply by r squared, I get r to the first power. I take a second derivative of that, I get 0. Okay, what's troubling me all of a sudden is, but I should be able to add a constant. Yeah, the r was for cylindrical coordinates, you're absolutely right. So what have I done? I know I can add a constant. So there's something I have to go back and carefully go over the steps to make sure I get this right. But the idea is, in a spherical, and by the way, you can check if you really want to, that if I say this function of r is some constant times 1 over r, and I use r to be square root of x squared plus y squared plus z squared, you can actually do all these derivatives and show that, yes, indeed, it gets knocked to 0. Okay, so we have a small technical hitch here. What I'm going to tell you is that, all right, so let's go back here. Del squared C of r equals 0. That's the equation we have to solve for steady state. And I claim, and of course I have to justify my claim because I've dropped a stitch somewhere. You have the explanation?
I do not recall anymore what was really written there, but I think it was missing when there is the first, you derive with respect to r, then you have the factor r squared, and then you derive again. It's inside the parentheses.
Thank you. Right. In other words, so what I'm saying is that the solution is a constant. Now, obviously, constant is a solution of the equation. If you take any derivative of a constant, you get 0, and I'm trying to get 0 on the right-hand side. So yeah. Thank you. You have to distribute your derivatives and your r's properly. See, I haven't taught this kind of thing for a long time, so I didn't have that at the tip of my fingers. Okay, but the part that's interesting is that, of course, you can have a constant, but you can also have something that goes like 1 over r. All right. So why did I write C infinity? Well, okay. So or let's call it C naught. C naught is just the ambient concentration. It's what you get to when you get very far away. All right, good. And then there's this constant divided by r. Whenever you have a, so how do we fix this constant? All right. So we're now bumping up against the problem of finding the boundary conditions on a differential equation. So what are the boundary conditions? So now we have to talk about the boundary conditions. Okay, so the boundary condition is that. So we said that when I get out here, it's just whatever the environment is giving me is the concentration. However, when I get to the radius of the cell itself, what the cell is doing is absorbing everything that falls on it. That's the assumption. Well, if it's absorbing everything that falls on it, that means that right at the surface, the concentration is 0. Okay? So that's the idea. The boundary conditions are, when you go far away, it's whatever the ambient constant is, and when you're right at the surface, this guy is eating everything that falls on it. Stuff is not bouncing off. Once it gets to the cell, it'll find a receptor and immediately be eaten. And therefore, the concentration must be 0 right at the surface. Okay? All right, so that means when we put in our boundary conditions, what do they imply? Well, they imply that the thing is all right? In other words, this is the constant, and then this is the term that's going like 1 over r. And I've adjusted the constant so that when r is equal to the radius of the cell, I get 0. All right, so this is a so-called second-order partial differential equation. I need two boundary conditions. It's like Newtonian mechanics. It's a second-order in time equation. In order to get a complete solution, you have to give the initial position and the initial velocity. It's second order. All right, so that's the solution. That is the mathematical solution of the problem here. And what does that tell us? Well, okay, so why is this of some interest? Now, I won't ask, are there any questions about this? Well, I will ask, are there any questions about this? Yes?
If at this point I just wanted to consider that perhaps the cell has some finite efficiency with which it is absorbing these elements that are floating around, I'm just changing these boundary
I
conditions.
could do that. I could do that.
Right?
But I'm going to give the most straightforward solution is to say this cell is ravenous, right? It is absolutely ravenous and is therefore eating everything that it can, all right? So it simply immediately eats up everything that gets within a molecule's distance of it. Now, of course, there are other boundary conditions that could be imagined, right? It could be only partially efficient. That gets more complicated. There are other boundary conditions that I could imagine, okay? There's a story about Heisenberg and his computation of the amount of energy that could be released in a fission bomb, and he miscalculated. He used the wrong boundary conditions for thermal diffusion of neutrons, and so. But I won't go into the story here, but it is entertaining. Yeah.
Hey, are you considering that the particles there are entering freely the membrane of the bacteria?
Okay, so say it a little bit more precisely or slowly.
It's passing through the membrane without a receptor or something?
No, in other words, the assumption is that as a particle undergoes its random walk, as soon as it hits this boundary,
It's going to enter.
gone, okay?
Okay.
So that means that, when you're within 1 epsilon, 1 molecular diameter of the surface of this guy, the concentration has to be 0, because if it weren't 0, well, that's the way of translating the idea. Everything that falls on the surface is immediately eaten, translated to the
And
inside.
the opposite is true inside the bacteria? If
Well,
some-
inside, okay, so this is assuming that the bacterium never gets saturated or satiated, I should say. It just keeps eating.
Okay.
So it's just a model, simple model, but this is the maximum rate. This is to determine the maximum rate at which it can feed itself, okay? Okay, good. And what good does this, okay, so how do I get to the maximum rate at which it can feed itself? Well, the fact that the concentration is 0 right at the surface of the cell does not mean that the flux into the surface is 0, all right? So let's look at that. So the thing we're really interested in is flux into cell, okay? Now, I erased that. So the flux thing was times gradient. So the flow, you know, how many molecules per square centimeter per second is determined by this diffusion constant. That we have to measure, times the derivative, the spatial derivative of the concentration. All right, so now, I wrote this guy out explicitly in terms of ddx, ddy, and ddz, but I've kind of left ddx, ddy, and ddz in the dust, and I'm talking about derivatives with respect to r. So I can transform this guy into, okay, so in spherical coordinates, there's the direction that takes you in and out along the radius, and then there are the directions that take you in angle, right? So mostly on the surface of the Earth, we are only moving an angle. We're staying at a fixed distance from the center. But what the flux in the direction of the radius, so the flux in and out, the flux in the r direction is going to be the derivative of the concentration in the r direction. And that now I've solved for that, okay? Because, or I can write it down explicitly because I know what is this? Well, C of r is C naught times 1 minus A over r. And I differentiate. And so I'm going to get d times C naught times, oh, in fact, of course, evaluated at, for us, it's going to be at the surface of the cell, right? Okay, so at the surface of the cell, although the concentration is 0, the derivative with respect to where you are is not 0. And I'm going to get, I differentiate this, I'm going to get A over r squared. It would appear, and I evaluate that at r equals to A, and so I'm going to get d naught, d times C naught times 1 over A. Okay, so that's the per unit area, right? And okay, it's just some numbers at the moment. So now the total rate at which the cell can feed itself, total flux, or total rate. That's going to be the area, 4 pi a squared, times this thing we just calculated, right? So I get, when all the dust settles, there's a 4 pi, there's a d, there's an a. And so I can, once I know what I'm eating, once I know which molecule I'm talking about and can put in a particular value for this diffusion constant, and I tell you what the situation is, what is the actual concentration. This is a number, it's in molecules per second, okay? And all right, so let's see what I tell you about that. All right, so for reference, here are some values. So we have A is about 10 to the minus 6 meters, 1 micron. D, all right, for glucose, just as an example, it turns out to be 2 times 10 to the minus 6 times 2.6, excuse me, times 10 to the minus 6 meters squared per second. Okay, I mean, I'm using systematically MKS units, so all of these exponents look a little bit odd. Usually if you know in what domain you're going to be working, like you're talking about bacterial cells, so the distance scale of relevance is a micron, et cetera, it makes sense to use different units, to say A is 1 micron, and that this is, so this would be 10 to the 12, so this would be 2 point, this would be 1,000 micron. No, it's 10 to this. Sometimes we want to re-express these things in terms of microns rather than meters, but this is what I've done. Okay, we'll stick with it for the moment. And, you know, so now you have to talk about what can you get for glucose, and so what is the concentration of interest? Well, you get to control that. Let's see. In water, it's standard conditions, I guess. This C infinity is going to be 6 times 10 to the 3, 6 times 10 to the 23 per cubic meter. And we can work out how many, and I guess total flux. All of this means total flux is something like twiddles 10 to the 10 per second. Okay, you know, unless, I'm not going to try to go too deep into this. There are some interesting books that are worth looking at where a physicist and a biologist put together numbers. You know, what is in fact the rate at which a bacterium consumes oxygen molecules or glucose molecules or whatever. And you know, so these numbers that I'm getting for you they're numbers that don't convey very much information at the moment. I just wanted to familiarize you with the notion of diffusion setting up a maximum rate at which things can happen, all right? So this number right here that says diffusion constant times the concentration at infinity times a linear dimension, right? This number doesn't get bigger as the square of A, or smaller as the square of A. It only goes as the first power, all right? So some of your intuition about how rapidly things can happen, or what happens as you change the scale, is in fact not correct, right? Diffusion has some rather interesting and surprising consequences. Okay, now where I was going to go with this was, yes, okay, so now I'm going to step back a little tiny bit and perhaps say, I'm going to improve my model a little bit, and I will not go too deeply into the details. So this is how rapidly a cell can absorb stuff coming into it by diffusion. So let me now make this a little bit more complicated. So what is really going on? Well, we have this cell, and it's not eating over its entire surface. All right? What it's actually doing is, as I was saying before, it's capturing stuff with these little molecules that are quite a bit smaller than the cell itself. All right? So you might think that we are, well, we are to a certain extent fooling ourselves. We're imagining that the surface of the cell is completely covered with these receptors that are eating the molecules as they arrive. So the next improvement, if you will, of the model that is discussed by Berg and Purcell is, well, let's suppose that what's really going on is that although we have a cell that's got some size, it has little patches on it, little disks. And these little disks are what's doing the eating, okay? Because that is a more realistic statement. You don't have, I mean, we can work out what is the size of these little receptor molecules, you know, what kind of cross-sectional area do they have? Well, you know, they're proteins. They're of some standard size, and we could work out how many of them are actually needed to cover the surface of the cell, and it would turn out to be a completely absurdly large number. It doesn't, they don't cover the surface of the cell. So there's some number of little patches that have some size, radius S, quite a bit less than the size of the cell itself. So the real problem we have to solve is what is the rate at which a little patch, assuming that the patch is a complete glutton, it absorbs, it just eats completely anything that touches it, all right? But it has a size that's smaller than the size of the cell itself. So the idea is what is the absorption rate of a disk. So this is a little patch on the surface of the cell. Okay, so now those of you who've taken undergraduate physics will have spent some time, I hope, solving potential problems. You know, what's the potential of a circular plate? What's the potential of a sphere? Blah, blah, blah. If you put it at a certain voltage, how much charge is on it, and that sort of thing. Solving electrostatic potential problems is, in fact, the general theorem. It's the general problem of solving the same equation we were just solving with, you know, a different physical interpretation, okay? So in the Berg and Purcell paper, they essentially say, well, look up in your undergraduate text what is the capacitance of a disk. And that can be translated into what is the rate at which a disk patch on the surface of the cell. That will tell you what is the corresponding answer to the how fast can it eat problem. And, you know, the result that they get is pretty much the same. There are some pi's that are missing, all right, because you're going from a sphere to a disk, and when you do the actual mathematics, you will get some slightly different numerical formulas. So the absorption rate of a disk, the max rate, you know, is going to be max rate is for a cell where's my disk? Here's my disk, my one-sided disk. It's 4 times S times D times C naught. All right, so it's very similar in form to the thing that we could solve by doing a really elementary calculation. You know, there's a pi missing. Okay, big deal. Now, so if this patch, if the single patch is much smaller than the size of the cell, then the rate at which it's eating molecules that are diffusing in is going to be correspondingly smaller. If S is very small compared to A, the overall size of the disk. So probably you want to have multiple patches. So we have N disks. And then they go into the interesting exercise of, okay, if solving for the rate at which a disk can eat is a simple electrostatic potential problem, and I know how to do it, and I get an answer, what happens if I put N of them there? And how many of them do I have to put on in order for the total rate of eating to be equal to what I would get if I simply completely covered the cell, right, with patches? So I won't go there, but they're going to, what they find is that, let's see, so let's vary. So we have N disks, maybe we don't need this. I mean, I think this is conceptually interesting, but it's a little bit too complicated for me to give you the entire detail right now. Am I running out of time, Nathan? What is the? Oh, okay, it goes on forever, great. All right, so if I have n disks, they actually get us a formula. And they say that it is, so the electrostatic model, it goes like, is what we got over there. Somehow the pi crept back in. N times S times A over NS. So this is actually the end result. So dimensionally, it's all okay. Remember, 4 pi dc infinity C1A. No, sorry, J max. And J max is NS. So if n is 1. Okay, so this is what you would get if your spherical cell of radius A were covered, were completely covered, all right? But in fact, we just have n disks that are of a much smaller size. So dimensionally, this should be okay. If n is very small, it should be this, the formula for a single disk, which it is, because A is very much bigger than S. So if n is very small, I divide out the pi and I divide out the A, and I just have S in the place of the A, and I have n. So if I have several disks, I get several times the individual disk absorption rate. But if n gets to be very big, well, if n gets to be very big, it goes back to that formula. And you can ask the question, okay, so how many of these disks do you need in order to get, let's say, 50% of the maximum rate that you would get if you were absorbing all over the surface of the cell? And it turns out that number is about, so, okay, n equals what to get 50% of max? Okay, this is the max. This is if the whole surface of the cell is eating the molecules as they diffuse in. So that's big. And so how many of these guys do you need? Well, it turns out you need something like pi over 4 50 equals times S, sorry, times A over S, A over S, A over S. It's something like this. Oops. So this is actually a number that, let's see, what do we need? We need something like, how many needed for 50%? Something. Right. Yes, so this turns out to be about, if you take some reasonable number for the size of these patches, the size of a typical medium-sized protein, and this is the typical size of a bacteria, this is a micron. So this, there's a factor of 1,000. So it's 1,000 of these little tiny disks displaced over the surface of the cell gives you the ability to absorb almost as much as you could if the thing were completely covered. And 1,000 of these little guys, the total area that they actually cover is a very small fraction of the total area of the surface of the cell. Okay? So the physics of diffusion is a little bit counterintuitive. In other words, it's hard to believe that if you completely covered the surface of the cell with absorbers, and if instead of completely covering the surface of the cell, you just covered a small fraction of the cell with absorbers, you do just as well. Okay? And this is good because 1,000 sounds like it's a large number, but in fact, these are small objects, so you just have the surface of the cell decorated with a reasonable number of these receptor proteins, a number that's easy to make, right? It doesn't cause the cell to devote its entire ability to make proteins to making these absorber proteins. So diffusion plays a very, very important role in so to say, the energetic economy of cells. Okay, so the only thing these statements over here are things that you can extract from reading this paper, and depending on how deep your background is in physics, you'll find it more or less easy to follow. But I've tried to lift out what are the important things. Okay, so this is the beginning of the discussion of the role of diffusion. Okay, and here it was talking about how fast can a cell pick up molecules from the surrounding, supposing that it needs these molecules to eat. All right, now there's a different take on this whole problem that I want to start to get into. So let's see the various numbers to put in. What are the questions? Basis of diffusion, da, 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 da. All right, so much for eating. Now, in fact, if you look at what bacteria do, and what E. coli do, you find that almost like molecules, if you have a solution, you've got E. coli bacteria in it, and you're observing it through a microscope, you will see the E. coli bacteria engaging in what looks like a random walk. Now, this is not the random walk of molecules. It's not thermal motion. They're actually propelling themselves. They have little flagellae, and the flagellae, they have a screw sense to them, and if they turn one way, the cell is just propelled forward, and then every now and then, all of a sudden the cell stops and it just does some crazy dance, and then it goes off in some other direction. But if you sort of follow the whole thing, what you see is it goes in a straight line, then it sort of randomly chooses a new direction, then it goes in a straight line, and after a while it changes a new direction, and so on. So it looks a little bit like a random walk, but it's not the thermal random walk. So what is it doing? Okay, so in order to eat most effectively, you have to find where the source of food is. So in addition to extracting molecules from the surrounding medium, the cell is actually measuring, it would appear, that the cell is sensing where to go to find the most molecules to eat. Because, in real life, the things that the cells need to eat are not uniformly distributed throughout the environment. There's more here and less there and so on and so on. So in addition to actually extracting molecules, it has to measure. It has to measure where the molecules are. Well, it should measure what is the density of molecules in its immediate environment, and if it's not enough to be worth sticking around and eating, you have to go somewhere else. And you have to direct where you're going. You have to have some method of both measuring what's the density of things you're interested in here and going somewhere else and measuring again. And if you found that the density has increased, that's a good thing. you might want to continue doing what you were doing. And so there is a problem. So this is a sort of generic cellular problem. How to measure. X. So X might be glucose, but for us, you know, there are many circumstances in which molecules basically receive signals from others, sorry, when cells receive signals from other cells. So the cell emits some small molecule, or maybe can even be a protein, and it makes its way to some other molecule, and if that other molecule senses its presence, so if the other cell senses the presence of this signal, it can do something. All right? So signaling is an extremely important aspect of living cells, and in order to, you know, they have to both emit signals, and the signals are, except for the case of nerves, the signals are molecules or proteins, okay? And they go from one cell to another, and the cell that is receiving the signal has got to have a device for sensing. Well, the basic machinery is the kind that we've been talking about already. On the surface of the cell, there are proteins whose whole function in life is to detect specific signals, and then when they get the signal, they cause something to happen inside the cell. They might cause some new kind of protein to be read out from the genome or whatever. But I mean, there's the basic signaling problem. So, all right, so measure concentration of X. So X might be food or X might be a signaling molecule, all right? So persistent presence in your neighborhood of signaling molecule that says, I am being attacked by a virus. If you're a T cell, let's say, please turn on and do something, okay? That's a kind of signal that you want a cell to be able to respond to. All right, so we're now going to talk about signaling. And so the general idea is, here's my cell. It is decorated with some number of surface molecules, and these surface proteins, and their role in life is to detect the presence of some kind of signal. This is totally generic, okay? All right. Now, to go back to the question of eating, in order to know where to go to find a better restaurant, you might say, oh, it's easy. All the cell has to do is to measure the gradient. That is to say, now I leap back to the question of getting food. If the cell has receptors that can measure the presence of glucose at different positions on its body, if for a single such, if these individual receptors can actually measure the local concentration, as we say they do, you know, the rate at which they're receiving molecules of interest is something that is determined by the local concentration. All it has to do is to measure the concentration at the front and compare it with the concentration sensed at the back. And if it finds that, okay, the concentration here is bigger than the concentration there, then, you know, get your little flagellae in motion and go in that direction. All right, so the other issue that these guys talk about is, given the statistical properties of diffusion, the fluctuations in diffusion, is it possible for a receptor on the surface of a cell to make a measurement of the local concentration that's accurate enough that by comparing the concentration at the front and the concentration at the back, you can say, oh, the direction you should go to get to a higher concentration is that way, all right, or that way, as the case may be. And it turns, well, okay, so that's the question that I want to address next. Turns out that's almost the same question as how do I reliably respond to signals, okay? So signaling and So we'll talk about the whole diffusion problem in that context next. Okay, so how does this work? Well, if I have a receptor, there it is. It's either in this state or in that state. In other words, it's either empty or it's got one of the molecules that it's interested in bound to it. That's it. That's all it can know, right? And so we're getting into the question of information in living cells. So what are the data that cell has available to it to decide what it wants to do next? Well, either, so suppose at time t equals to 0, it's occupied. It has one of these molecules attached to it. Well, depending on how tightly bound this guy is, how big is the binding energy? What's the free energy difference between having this molecule in solution or having it out of solution and attached to this guy? There's free energy. There's a certain number of ergs or joules that is associated with that. Okay, the more tightly bound it is, the longer it will stay bound. But eventually it'll come off, all right? Because the binding energy is not infinite. It's not, okay, so I'm going to have a 1 and a 0. 0 is nobody's home. 1 is one molecule of the kind that I'm interested in is home. And so there's a time series to think about. So as time goes on, this guy will stay here, but all of a sudden he'll come off. All right, so this is going to go to this with some characteristic time. All right, the so-called unbinding time, which has to do with the strength of the binding energy. And we'll come to that in a second. So basically, all that happens is that, so this guy is bound, and then he comes off, and then after some period, a new molecule can come out of the solution and bind to it. And all that the cell has available to it is this time series. Something is bound. When this receptor is on, and now it's off, and all of a sudden it's on again, and now it's off, and now it's on again. That's it. That's all that it can know. There's just nothing else. So then there's a characteristic time here. And it's not that it stays on for this time. It's that the probability that it comes off, makes this transition per unit time, is 1 over this characteristic time. And then the rate at which, well, okay, so the rate at which new events can happen have to do with the rate that things are arriving from the outside, and we have some information about what it is that governs that. The diffusion constant and the concentration governs that. So this is what the cell has to work with. And so basically we'll call this P. So it has this time series. And the question is, what can you do with that time series to, all right, so what do you do with this time series? That's the question. So we can find the average, the average occupancy. And the average occupancy will be bigger if the concentration outside is bigger, and if this unbinding time is longer, and otherwise. So what we'd like to do is to get some, so this is certainly one thing the cell can do. You can measure the average time that the receptor is occupied. So what I'll show next is that this is connected to the concentration outside. So this is proportional. It's proportional to C. Okay, so it's a good thing to measure because it's related to the concentration. Okay, it seems pretty plausible that that's going to be the case, and I'll give you the argument in just a second. In any case, if you know the average occupation of the receptor, that gives you access to the concentration outside. So you say, fine, very simple. I have different receptors on the surface of the cell, and therefore all I have to do is to compare the average, the measured average occupation number at the front of the cell and the back of the cell, and if there's a difference, then I know I should go in that direction. However, all of biology is about statistics, right? So in point of fact, that's fine, but the thing you really have to be concerned about is what is the variance. Okay, what's the variance? Because if the variance is too big, then the accuracy of your measurement that you extract from one of these guys is not going to be enough for you to reliably say who's bigger and who's smaller, right? So the other thing you need to know is, and what is this? And how does it compare to that? And if you know, so what we'll do is to go through an argument in which we say circumstances are such that there is a limit, so this will limit the accuracy of measurement of C. So our concern is going to be what is the ratio of the variance to the mean? Delta C over C is going to be the problem. And if it's too big, then we're in trouble. All right, so we really have to know what to do. Now, those of you who know a little bit about statistics will say, it's easy if you just measure long enough, if you take enough independent measurements. If this guy is fluctuating, all I have to do is to take n independent measurements of the mean to be able to get a global measurement whose variance is reduced by a factor of square root of n. All right, so this is the usual data analysis thing. And so that has to do with time. How long do I have to measure in order to get a measurement of C with a certain accuracy? That's the thing. All right, so this is what these guys also talk about. And I think it's a really interesting, intellectually interesting exercise. So let's go through that. All right, so, and the thing is, it's telling us something that's important not just for figuring out how bacteria can find out where to eat, but it also has to do with what does it take, what are the conditions on turning on and turning off vital functions in cells, like deciding when to die, right? There's apoptosis, which is very important, and that too has to do with signaling, and you have to have a reliable measurement of the signal that says it is time to die, right? And if you don't die, you're going to be the source of a problem, or it's time to turn on the immune system. So all of those questions, which are really fundamental to multicellular biology and medicine ultimately, have to do with this little statistical problem here. So I think it's worth carrying through this exercise. It's done in this paper, and I think if you read it straight, you will be totally confused. Maybe I can walk you through the major steps so that you will be able to see, roughly speaking, what the strategy of the argument is and figure out how you could actually reproduce it yourselves, okay? All right, so how am I doing on time? 5, okay, so we'll set up, the first part of this is, okay, what do I learn when I learn what is the mean occupancy of my receptor, okay? Okay, learn from, all right, learning C from P bar. Okay, so there are two ways of getting at this. I would say the most straightforward is kinetics. The less straightforward, but also very important, is basically Boltzmann Statistics. Okay, so I think I will be able to do both, but it's important. I think the simplest one is this. Okay, so again, we're going to assume that we're in steady state. All right, so all fluctuations have died out. We are in a situation in which we've observed enough to see things going on and things going off. And on average, it's got to be the case that the rate at which on goes to off, the rate of these events, has got to be equal to the rate at which things go on. That is to say, these events. From off, I go on, all right? So what is the rate at which I can go on? Well, I can't go on any faster than things can diffuse in from the outside, okay? So I must multiply this famous number. So this is the rate at which things fall onto a disk of radius s, given that the outside concentration is c, and the diffusion constant for the thing that I'm interested in is d. Now, if the receptor is already occupied, it can't, this new guy diffusing in cannot go on. In other words, for these receptors, the assumption is that you can bind one thing. That's it. Now, you could imagine a situation in which this receptor could bind 1 or 2 or 3 or 13, right? That's a possible extension of the idea. But let's assume that this is, there's just no room to bring in somebody else. This guy either is occupied or wants or not. So this is P average. Therefore, the rate at which things can go on has got to be it's got to be the rate at which they are arriving. And this we just derived, okay? So remember, this is diffusion constant, which has length over time squared. And this is the size, that's the length. And concentration is 1 over length cubed. So with any luck, LT squared. Yes, and sorry. Diffusion constant has dimensions of length squared over time. And so when all is said and done, this whole thing has the dimensions of 1 over time. And this is 1 over time. Okay, so we're good. All right. So we have this average occupation number, P bar, and we have the concentration outside, and then we have numbers that are fixed, right? My receptor has a certain size, that doesn't change. The diffusion constant of the signaling molecule is whatever it is. I mean, this is a specific signaling molecule. The only thing that can change is the concentration, right? The signal is either there at very low concentration, or it's there at an intermediate concentration, or it's there at a higher concentration, okay? But what this does, on rate has got to be equal to off rate in steady state. It tells me that, okay, I can solve it. I can solve it for what is the average occupation or I can solve it for I guess the other way of looking at this is all right, so I should have written this first as P bar over 1 minus P bar is equal to 4d S tau B times C. All right, so if you measure this guy, then all of this stuff, which is, it can be many different things, but it's fixed. this allows me to measure C. So this is how knowing the average occupation of the receptor tells you that the concentration is such and such. Now, the only thing that is missing here is that it's nice to know what is this off rate, so-called, in terms of some energetic quantity. All right, so I guess I can't do it this time, but maybe next time I can fill it in. So statistical mechanics allows me to determine okay, this quantity right here, I'll call it C half. All right, that's the concentration in which the average occupation number is 1/2, all right? It's equally probable to be on or off. Okay, so the statistical mechanics allows me to say that C half, which we know, right? I mean, we have from this formula, we know that it's 4ds tau b. Statistical mechanics arguments will tell me that this is actually e to the beta times the binding energy. Okay, so the point that I want to make is that there's an energetic thing. That is to say, if the signaling molecule comes out of solution and winds up bound to the receptor, that's a change in free energy. That change in free energy is the thing I call epsilon. That's the binding energy. You know, a tenth of an electron volt, half an electron volt is some number, right? But it's in the chemistry and atomic physics range. It's fractions of an electron volt. Beta is 1/kT, right? And this thermal wavelength, well, I can write it, it depends on, how shall I put it? It's got dimensions of length cubed. It depends on h bar. You know, quantum mechanics comes in here, all right? But the thing that's relevant for us basically is that this C half, or the binding time, the average binding time, is exponentially related to the binding energy, all right, associated with taking that signaling molecule out of solution and letting it attach to the receptor. Okay. It's good to know this, and we may make use of it, but the main point is, okay, if you measure the average occupation probability, you're actually measuring the concentration. And, okay, so it's at this point that we get to be concerned about, and what's the variance? What actually determines how accurately you can determine this average? Okay, and so that's at the heart of some important aspects of actual biological behavior that you can't understand unless you really understand how diffusion and statistics work here. So we'll follow up next time and figure out how to connect this variance to the concentration, the time that you take for your measurement, and all the rest of that stuff. And there's a moderately complex set of steps in between, but when you get to the end, the result is really quite simple. And it illuminates a bunch of things that one actually observes in bacterial behavior and in other signaling situations. Okay? All right. Until tomorrow. Okay, so by the way, I'm going to try to figure out how to post stuff for you so that you can download it, and I hope that there's some, so to say, email mechanism for communicating with you, because I guess I'll put this on Dropbox and I'll give you I'll email the link, and I'll work on putting stuff up there seriatim. I'll put up this paper and maybe some notes of this kind this afternoon, I hope. Okay? So, I mean, I guess the real question for you is, am I talking at a level, you know, am I doing this at roughly the right rate and level of assumptions of what you already know? Am I boring you? No, not boring you. Okay, good. Is it all just, I mean, usually people do this kind of thing. They prepare some PowerPoint slides and they flash the slides past you. And my impression is that the effectiveness of that for communicating the essence of this kind of subject is very low, right? So I'm going slowly, but I'm actually, as I say, opening the hood, showing you where the spark plugs are, unscrewing one or two of them, putting them back in. But I'm not doing a complete disassembling of the car and reassembling it, but I'm giving you some sense of what it is you have to do in order to go through the steps to really understand it. But there's absolutely no substitute for just going through it line by line yourself. And there are parts of this paper, and I'll try to point them out to you, where they're telling you more than you need to know, and other parts where they're telling you things you really need to know. And I try to give you some guide to this. Also, there are textbooks, 1 or 2 textbooks that cover this kind of thing in this general style, and I'll give you the titles and authors' names and so on. Okay? All right. Until tomorrow. Okay. Okay.